Welcome, everyone, wherever you are joining us from, to today's Global Immunotalk. Global Immunotalk is a wonderful initiative. It's been running since 2020 to benefit and inspire immunologists from all around the world in an egalitarian and environmentally friendly manner. Now, I am Caetano Reis Souza, and I have the honor and privilege to host today's Global Immunotalk speaker, Andrea Schietinger, who joins us to deliver a lecture entitled CD8 T cell fates in cancer and autoimmunity. Now, Andrea is originally from Germany, and she did her PhD with Hans Schreiber as a joint program between the LMU in Munich and the University of Chicago. She was then a postdoctoral fellow with Phil Greenberg at the University of Washington before moving to New York and establishing her own lab at the Sloan Kettering Institute in 2015. And since 2023, she has been a full member of the Institute. Now, Andrea's career focuses primarily on the regulation of CD8 T cell responses to cancer and more recently in autoimmunity. And he has made key contributions to our understanding of how CD8 T cells are subverted in cancer and how they can be reprogrammed for therapeutic purposes, but also an interest in autoimmunity, and has recently described how this can be sustained by stem-like CD8 T cell populations in the affected tissue. So as usual, today's talk will be followed by a Q&A session in Twitter/X, in which Andrea will endeavor to answer your questions over the next few weeks. We will have instructions on that right at the end with a slide that directs you to the right page to do so. So just before I invite Andrea to deliver her lecture, I will start by asking her, as it is usual in this series, ask her a question about herself and her career. And the question, Andrea, for you is, what is the advice you would now give to your 25 to 35-year-old self?
So first of all, thanks for having me, Caetano. Thanks for the kind introduction, and for the organizers to giving me an opportunity to participate at this amazing lecture series. So the answer to that question is love what you're doing. Science is hard enough, but you really need to really be passionate and love what you are doing. Don't take rejections as a personal reflection on your science, because as a postdoc, as a junior faculty when you're establishing your lab, there are many roadblocks that you have to overcome, and you always have to think about the ultimate goal in life is to really do great science. Take your time for it. Try to be right and not first. And yeah, I think really to understand that you're in for the long run, and you need to overcome these sometimes really hard rejections. And I think that is something that I wish I could have get the advice when I was a junior faculty, to really realize this is just part of the game, to have these defeats, and you just keep going with your team, and you celebrate every single success in the road of doing the science.
Well, thank you, Andrea, for those very inspirational words. And so I will now invite you to share your screen and deliver your lecture.
Okay, so let me share. Okay. Oops. So everybody can see that as a full screen. So like I said, I'm really honored to present this lecture today. Welcome everybody around the world. And what I'm going to talk to you today is our work we have been doing over the last 9 years here at Memorial Sloan Kettering, summarizing maybe the the first few years of really looking specifically at CD8 T cells in tumors, why we started thinking and working in autoimmunity. And at the second half of the lecture, I'm going to present a new unpublished story on the role of CD4s and how they are contributing in defining the fate and reversing the fate of T cells in tumors. So for those who don't know me maybe that well, we are a lab that is really trying to understand what are the factors that really determine how a CD8 T cell differentiates. What is the contribution of the factors that drive T cells to become either functional or dysfunctional? And as you all know, the context in which a naive CD8 T cell encounters antigen and gets activated really defines what the fate of that T cell is. And there are different scenarios we are studying in the lab as a tumor immunology lab. We are obviously interested in understanding why, when, and how CD8 T cells in tumors derail. We have genetic mouse models in the tumor context. We are comparing it to settings where things really work extremely well. For example, in infection, acute infection, where you have naive pathogen-specific CD8 T cells, they become highly functional effector T cells, ultimately form memory. How is this T cell differentiation contrasting to the T cell differentiation path of a T cell within a tumor? But then more recently, we have been really trying to understand what actually kicks off self-reactive T cells to suddenly attack normal tissue. I will tell you how and why we started thinking about this and what the interesting features I think are that help us to understand why things are derailing in the tumor context. So as I said, as a tumor immunologist, we think about what are the factors that really drive CD8 T cells in a dysfunctional state. And when you think about T cells in the tumor context, you think generally first about T cells sitting in an established tumor, and there are many immune suppressive microenvironments that contribute and inhibit CD8 T cells from working, and they're listed in this box here. But this is end stage, so to speak. A tumor doesn't develop in a short period of time. There is a long process that leads ultimately to the establishment of a solid tumor. And generally, tumors are a clonal disease. You start in normal tissue. You have a malignant cell that potentially can express unique antigens on the surface, an oncogenic driver or so forth. And our question was initially, when does actually T cell dysfunction start? When do you have the earliest features of T cell dysfunction in a tumor reactive T cells over the course of tumorigenesis? And this is the question that we really started to address when we started the lab here at Memorial in 2015. What is the fate of a tumor-specific T cell that encounters antigens, specifically at the earliest stages of tumorigenesis? And so now I'm going to summarize the work we have been doing in that space. We used genetic mouse models, autochthonous mouse models, where we can induce in normal tissue an oncogenic events, and then we followed the fate of these tumor reactive T cells that really are specific for the transformed cancer cell. And what we found was that already at the earliest time points of tumorigenesis, T cells, in this case, it was an autochthonous liver cancer models, they infiltrate into the malignant lesions, pre-malignant lesions. They get activated, but they actually get very activated in the wrong context. And so Mary, Andrew, and Moshta, they were previous trainees. Mary has now her own lab at Vanderbilt University. And these fabulous people really contributed to understanding where and how these T cells derail. So what we found was that after the T cells encounter the malignant cancer cell, at these earlier stages, they already become dysfunctional. They cannot make effector cytokines, and that allows really the tumor to continue to grow. We found that there are epigenetic programs that are induced through this unproductive encounter of the T cell with the cancer cell. And we actually distinguished two phases of T cell dysfunction. We found that this is a progressive differentiation process that starts with an early dysfunctional state that has a very unique epigenetic program. And early on, these T cells are reprogrammable. So you can certainly with, for example, immunization or checkpoint or other immunotherapeutic intervention, put these T cells back into a functional state. But at some point with time, these T cells enter a fixed state that is resistant to reprogramming. And we found that over time, T cells in progressing lesions lose an important transcription factor you're going to hear a lot about today. It's called TCF, which is a hallmark transcription factor associated with stem cell renewal, memory formation, and so forth. And what we found was that over this period of tumorigenesis and T cell differentiation, a T cell loses that important transcription factor with time. What we also then found was there is a master regulator transcription factor that is responsible for pushing these T cell into this dysfunctional fixed state, and that is TOX. And I'm going to just very briefly talk about TOX, what we think TOX is actually doing in T cells and contributing to the phenotype and to the functional state. And so this was our work, but Mary followed up, and I want to just highlight a really beautiful paper, a follow-up paper, where she actually really looked at the precise timeline of when you see the earliest features of T cell dysfunction in a T cell that encounters the cancer cell. And what she found was that it is really within hours where the fate and the commitment of a T cell in tumors is determined. And so with that, with this work, we were interested in understanding what are the key transcription factor that pushes naive T cells into this plastic and fixed dysfunctional state. Is there a hallmark transcription factor, and what is this transcription factor responsible for? So we were thinking and hoping we would find a transcription factor that really is responsible for driving T cells in the dysfunctional state. And so one of the requirements was to say this transcription factor must be probably then uniquely expressed in dysfunctional T cells while they differentiate in the tumor context versus in a context where you really produce functional effector and memory T cells. And so one slide I would like to present the story on TOX because it will be important later on in the autoimmune context. So as I said, we were trying to understand and identify a transcription factor that is responsible for this epigenetic driving of T cell dysfunction in tumors. And most of you have heard now, this is the transcription factor TOX that we and others identified, for example, also in chronic viral infection. There have been beautiful papers. This is a transcription factor that is highly expressed in these T cells that continuously encounter antigen. And we looked in the tumor context, and because you do have T cells sitting in these malignant lesions, getting stimulated in the wrong context with antigen, that drives the expression of TOX. That correlates with the high expression of numerous inhibitory receptors and actually with loss of this transcription factor TCF1. And so we also looked in numerous human tumors, and we found exactly the same phenotype, that the T cells that expressed high expression of TOX were also very high expressing inhibitory receptors and activation markers PD1 and 39. So we got at that point very excited because we saw this is exactly what you want to look for, a transcription factor that is selectively high expressed in this dysfunctional T cell, because we argued, or we were hypothesizing, all what it takes is to knock out that transcription factor, and then you probably restore or prevent the dysfunctional phenotype of the T cell. And this was really a very surprising finding that really got us to think about what T cell dysfunction and some people call it T cell exhaustion, really defines in a T cell in the tumor context. So what we did is we knocked out this transcription factor tox in tumor reactive T cells. So here you see in red your tumor specific T cells that cannot express tox because it's genetically deleted, and you compare it to wild type T cells that are tumor reactive. You put them into a mouse that expresses a tumor, and then we look for the phenotype. And this is where we got very excited because, as you can see, the T cells that do not express tox, they look completely normal, similar to a naive or effector-like T cell. They cannot upregulate canonical inhibitor receptors, PD-1, LAG-3, and so forth. They look phenotypically very, very functional. And we looked transcriptionally and epigenetically, and we found exactly the same thing. Where the most surprising finding then came was when we said, well, now let's look at these phenotypically functional T cells. And what we found was that they are absolutely dysfunctional, similar to the wild type tox expressing T cells. They cannot produce interferon gamma and TNF. So this was the first surprising finding that tells you that the phenotype is not defining the functional state of a T cell, because I think in the field of tumor immunology, we look for PD-1 and we actually associate this with a dysfunctional state. And that is actually telling you that not necessarily is always the phenotype associated with the functional state of a T cell. So then the second surprising finding was that these tox deficient T cell that looked completely normal actually got lost with time. They did not persist in the tumor. So now how do you put all these findings together? We have done more characterization and functional studies, and I'm not going to present it because I really want to move to the newer and unpublished work, but it is important because what we now interpret phenotype and molecular features and functional state of T cells is, I think, important for us to really think about, okay, how can we reprogram T cells in the tumor context? So I'm going to summarize now here what we in the Schietinger lab think tox-induced dysfunction in T cells in tumors is all about. So you have a tumor reactive T cell in a tumor microenvironment. They see antigen in the beginning in an unproductive manner, but this becomes then a really continuous stimulation. And through this continuous antigen encounter, you do have chronic TCR stimulation. And that drives the expression of tox. And tox is responsible for the upregulation of numerous inhibitory receptors. So when I started first with my team to really delve into the role of tox and so forth, what I was under the assumption is that these are all inhibitory receptors. They're bad for a T cell, and that's why we are targeting it in the clinical setting. But when you actually look at the earliest papers that were published on the role in the characterization of these inhibitory receptors, such as PD-1 and LAG-3, you do recognize that these are not just inhibitory receptors. These are activation markers. And so every single T cell that you activate, including, for example, in vitro with antigen or anti-CD3, will upregulate within hours high levels of PD-1 and LAG-3. So these activation markers are molecules on the surface that tell the T cell to not get overstimulated and to prevent activation-induced cell death. And so this is a physiological negative feedback mechanism that prevents T cells from getting overstimulated when they encounter antigen in any kind of context, not just in a chronic, but also in an acute in vitro activation context. And so we thought that that these inhibitory receptors ultimately inhibit effector function. But what we found was, instead of, when you knock out tox, you lose this expression of these important inhibitory receptors. Now there is no safety mechanism. The T cells get overstimulated. They undergo activation-induced cell death. And that's why we see this loss of tox deficient T cell in the tumor. So we actually think that tox is absolutely required for T cells to persist in tumors and not to undergo activation-induced cell death. So tox drives this exhaustion or dysfunction phenotype in order to persist in tumors. And this is the module 1, we call it in the lab, that is downstream of the TCR, really defining the phenotype that allows T cells to persist. But downstream of the TCR, there is a second module that is really associated with the loss of effector function, and we think these are uncoupled. So what are the effector molecules? This is interferon gamma, TNF, FAST, trail, granzyme, perforin. And so now what we need, I think, to do is to understand, so what regulates the transcription, the translation of interferon gamma and TNF. We know from beautiful work from Monica Walker's lab in the Netherlands that interferon gamma is highly post-transcriptionally regulated. So we need to understand why T cells that are potentially or that are dysfunctional and exhausted actually have higher levels of interferon gamma, but zero protein. What is the block in making actually the protein to be transduced? What are the post-transcriptional regulatory mechanisms? Then also granzyme and perforin in some dysfunctional exhausted T cells are pre-made. They are in vesicles stored in the cell, but they cannot get exocytosed. So what is this block of release and exocytosis that prevents T cells from really being cytotoxic? So we think that in a dysfunctional T cell, there are two modules. One is for the phenotype and for the persistent, and the other module is for loss of effector function, and they are uncoupled. So that will be important for the autoimmune context. That's why I spent a little bit more time of really getting that point across. So now at like 4 or 5 years ago, we were at this point, we identified this important transcription factor, tox. We know that the T cells are terminally differentiated. You cannot really reprogram them. They lost this important transcription factor, TCF1. So what are you going to do as a tumor immunologist to really reprogram these dysfunctional T cells in the tumor context? How do you make them functional again? Everything what we tried actually failed. So back then we were thinking, okay, so where are we turning, and what is potentially our role model of T cell that we have to engineer, that we have to understand and really produce for anti-tumor immunity? What is the requirement that a T cell has to bring to the table? It has to maintain effector function in this context of persistent antigen in the tumor. But where do you have this feature that you have a T cell that goes into peripheral tissue and gets presumably stimulated with persistent antigen, but does not become dysfunctional, but continues to be cytotoxic and sustain tissue destruction? That's an autoimmune T cell. So an autoimmune T cell is the self-reactive T cell that suddenly gets kicked on, goes to the peripheral tissue where it presumably gets stimulated with self-antigen in a continuous manner. And in this context here, you do not end up with a dysfunctional T cell, but a hyper or hyperfunctional T cell that sustains tissue destruction. So we were wondering, if we want to really successfully reprogram T cells for the tumor context, do we potentially need to understand autoimmune T cell differentiation, T cells that, how do they initiate and sustain tissue destruction? And so a fabulous student in the lab, Sophia, took on this question. She wanted to understand, where do autoimmune T cell responses come from? This was done with a great collaboration from numerous colleagues and friends. And so we wanted to use a clinical model, a clinically relevant model, where we have autoimmune CD8 T cell destroying tissue. And one of the models was the so-called NOD mouse, the non-obese diabetic mouse that sporadically, spontaneously develops type 1 diabetes. Type 1 diabetes is a CD8-mediated autoimmune disease. You have autoimmune T cells in the pancreas, in the islets, in the insulin-producing islets, and they continuously destroy in this model over many, many months the islets and the mouse becomes type 1 diabetic. From beautiful work from many other groups around the globe, what was known is that beta cell specific T cells get primed and activated in the lymph node, and then over time, through a stage of insulitis, ultimately become or drive type 1 diabetes. And so we were wondering, or Sophia actually then asked the question, well, how do autoimmune T cells get primed, activated? What sustains? How do they sustain tissue destruction and do not become dysfunctional? And so what she did, she used tetramers to identify beta cell specific T cell populations. In this case, she used IGRP, which is a really immunodominant epitope recognized by T cells in mouse and in humans. And she looked in the pancreatic lymph node and in the pancreas. And you see clearly there are very nice defined populations. And so we asked first, okay, how does an autoimmune T cell look? We looked at exactly the same molecules on the surface of T cells that we profiled in tumors. And to our surprise, we found that many of those inhibitory receptors that we associate with T cell dysfunction in the tumor context are actually also expressed on autoimmune T cells in the pancreatic lymph node and in the pancreas. And so they have clearly an exhausted-like phenotype. But when we took these T cells out and we restemmed them, that's where the surprising feature was. Despite the expression of these exhaustion markers on the surface, they were highly functional. They could actually produce high amounts of interferon gamma TNF. So now this feature again suggests the phenotype of a T cell does not define the functional state, similar to the data that I showed you in the tox deficiency. You can be very normal looking and cannot make effector cytokines. Now here in the autoimmune context, you have the reverse scenario. You express inhibitory receptors, but you are totally fine and functional and cytotoxic. So this unique feature of autoimmune T cell was really exciting for us. And we wanted to understand what are the transcription factors that really are associated with this phenotype and functional state. And I'm not going to go now into details. I come straight to the transcription factor that we wanted to then further characterize and understand. It was again TCF, what I told you is associated with memory, self-renewal, and stemness, because you can see that in the pancreatic lymph node, in these diabetic mice or in these NOD mice, there are actually two populations of beta cell specific T cell. There's one population that expresses TCF1 and another transcription factor, or another population that has lost it. And in the pancreas, mostly of these T cells are TCF1 negative. So we were curious to understand, well, what are the functional capacities and characteristics of these three distinct populations? So we isolated the TCF high from the pancreatic lymph node and the TCF low from the pancreas, and we did various adoptive T cell therapy, adoptive transfer studies, and one where we isolated these distinct populations and we transferred them into NOD-SCID mice. And what we found was that when you transfer these TCF high cells from the pancreatic lymph node, you actually induce disease in the host, but you can transfer many TCF low sorted T cells, and they don't mediate disease post-transfer. So that was curious and interesting to us, because how can a population mediate disease while the ones in the pancreas actually don't? What are the cell intrinsic features? And so what we found was that even when you do serial transplantation of these TCF high cells for over 7 to 8 months, in the third, fourth host, you always have onset of type 1 diabetes. So these TCF high T cells, we characterize them molecularly, more functionally. And what we now find is that these T cells have stem-like features, stem-like characteristic. they actually give rise to themselves, they self-renew, and they give rise to differentiated progeny to the TCF flow. And so the model that we are putting forward in the autoimmune context is that in the pancreatic lymph node, there is this autoimmune stem-like T cell, it self-renews. When you transfer them into not wild-type mice or not SCID mice, they self-renew. They really maintain a population that is TCF high, but they continuously generate TCF low differentiated progeny. And in this model of autoimmune diabetes, these TCF low T cells migrate to the pancreas and do the killing. And these TCF low T cells that actually go to the pancreas in the peripheral tissue, they are short-lived. And you need this continuous replenishment from that stem-like T cell population that maintains the continuous destruction of tissue in the periphery. And if you block potentially the migration or the egress from the lymph node, you completely prevent type 1 diabetes, suggesting that really the driving population that gives rise to tissue destructing T cell is really the stem-like population. So now, going back now to the tumor context, you have in the tumor context this continuous differentiation within the malignant lesions in the tissue. We learned from the autoimmunity that there's a population that is clearly tissue destroying, and the initiation phase is the key feature that potentially distinguishes these two contexts. Tumors originate from normal tissue, autoimmunity originates from normal tissue. So what is distinct in the tumor versus in the autoimmunity context? In the autoimmunity context, we have these autoimmune stem T cell. Now the question is, why are they not being generated in the context of tumorigenesis? And so now with this, we ask the question, well, what is the breakdown then potentially that does not allow T cells to form in the lymph node in a really productive way? So in situ during tumorigenesis, you have this naive T cell that get activated, and this is actually happening without inflammatory cues in a non-inflammatory context, without CD4 help. In addition, when the tumor then progresses, you have immune suppression in the microenvironment, you have persistent antigen, and that all drives ultimately the T cell to become dysfunctional, to express high levels of tox, to lose TCF. So this is in the center here in the box, this is your terminally differentiated T cell. But what never really made sense to me and to us when we thought about, well, yeah, there you have clearly an unproductive priming event that makes T cells derail very early on. So you never really go through an effector phase like in infection. But in adoptive T cell therapy, there is there you take tumor reactive T cells, whether they are TIL or you engineer them from scratch by transducing them with a TCR or with a CAR receptor. You really generate highly functional cytotoxic effector CD8 T cells, and you adoptively transfer them into a solid tumor patient or a patient with malignant lesion. And even in this context, despite that you give billions of highly functional cytotoxic effector T cell, adoptive T cell therapy with CD8 T cells is largely giving you very disappointing results. And so we asked the question, is there something that we are fundamentally missing in terms of what is required for a highly functional T cell, such as in the context of adoptive T cell therapy? What is the missing? Is there a missing factor that prevents T cells to really mediate cytotoxicity in peripheral tissue? And so now, coming from the autoimmunity field, we were actually looking at autoimmunity, and we asked the question, well, can we gain some insights from autoimmunity to understand and to identify what that missing factor is, or this signal that allows CD8 T cells to really be cytotoxic killer T cell? And so we know, for example, in type 1 diabetes or multiple sclerosis, these are clearly CD8 mediated autoimmune diseases where you have a pathogenic CD8 T cell going to the tissue and mediating the killing of target tissue. And so despite the fact that we know the CD8s are the killing T cells, there is a strong genetic HLA-2 association in type 1 and in multiple sclerosis, suggesting that CD4 T cells are actually critical somehow for tissue destruction. So in mouse models, published and unpublished data from us now suggest that the CD4 T cells are absolutely required for the induction, but also for the maintenance of tissue destruction, despite the fact that you have the CD8 T cells being the mediators of cytotoxicity. So therefore, we ask, well, what is the role potentially for CD4 T cells in driving CD8 T cell mediated tissue destruction? And this is work from Gabby, who was a postdoc and is now back in Barcelona to continue his career. So now you can say, well, CD4 T cells have a name. They're called helper T cells for a reason, because they have been shown to really be helper T cells that drive CD8 T cell function. And when you look in the literature, it's absolutely true. There is beautiful work on the role of CD4 T cells, helping CD8 T cells to get properly primed through dendritic cell licensing, co-stimulatory molecules, important signals such as CD70 and CD27 from the Borst group, beautiful work, and so forth. And other contributions of CD4s have been described by, for example, allowing CD8 T cells to go into peripheral tissue, that this is also CD4 mediated. And other literature has demonstrated that CD4 T cells actually can directly, in an MHC2 dependent or independent mechanism, mediate tumor elimination. But what we were trying to understand is, are CD4 T cells required during the actual killing phase in the tissue? And if yes, how? And so this is Gabby's work, where he then went with a very reductionist approach and said, okay, let's understand whether there is a contribution of CD4s, not just for the priming, activation, and allowing CD8 T cells to traffic to the tissue, but also during the killing mechanism within the tissue for cytotoxicity. So what he did, he engineered tumors B16 or MCA205, which express model antigens, CD8 or CD4s. In CD8s, we use Synfacl from OVA here in green, and then we used a CD4 epitope, GP61 from LCMV, and these epitopes are recognized by OT1 CD8s or by smarter CD4 T cells, and they are physically linked. So the tumor expresses CD8 and CD4 epitopes on the surface. And so we modeled a tumor model where we inject these tumors, we let the tumors grow, and then when they have a nice size after 2-3 weeks, we adoptively transfer CD8 T cells that have been activated in vitro, made highly functional cytotoxic CD8 T cells. And we adoptively transfer them, and this is exactly what you sometimes see in these, or what you mainly see in these patients with solid tumors, that when you give a large number of CD8 T cells that you generate in vitro, you do not necessarily get tumor elimination. So this was our starting point. And now in the second cohort, then we said, okay, now let's add the activated CD4 T cells. And what we found was 100% complete elimination of solid tumors and long-term protection from recurrence. So first we asked the question, so how is this working? We looked first at the numbers, because we thought, well, maybe the CD4 T cells are still helping more CD8 T cells to come into the tumor tissue. And when we quantified the numbers of CD8 T cells, they were absolutely identical. So we had ultimately in the tumor that progresses or progresses or gets rejected, equal numbers of CD8 OT1 T cells. So it's not a numbers game, But then when we looked at the function, that's where we saw huge differences. That the CD8 T cells that got the CD4 T cells, they actually were highly functional. They could make high levels of interferon gamma and TNF. But the ones that didn't have co-transferred CD4s, they were very dysfunctional. And ultimately, that led to the outgrowth of the tumor. So then we looked at numerous inhibitory receptors and tox expression, and you see these are much lower expressed when you give CD8 T cells, CD4 T cells. So clearly the CD4s cell intrinsically did reprogram the CD8 T cells in the tumor. We asked the question, is this just something very unique to the tumor tissue, or is this already reprogramming happening in the tumor draining lymph node? And so when we looked in the tumor draining lymph node, this is not data I'm showing, but we have the entire data set. There is absolutely no difference in the tumor draining lymph node between a CD8 T cell that saw or that received co-transferred CD4s or CD8. So whatever the difference was only generated and mediated within the tumor tissue itself. So then the next question was, okay, is this just a unique signal that is changed through the additional contribution of CD4s, or is this a global reprogramming? And just to give you a flavor of it, we did RNA-seq and ATAC-seq, and you see many transcription factors, genes, and signals being affected, and especially chemokine receptors and so forth. On an ATAC-seq level, looking at the chromatin structure, you also see huge differences. So we are talking here not about a couple of signals and genes that are being reprogrammed or different. It is really an entire genome-wide reprogramming that the CD4s do with the CD8 T cells. So then the question was, okay, so how do the CD4 T cells do this? How do they prevent CD8 T cell dysfunction? And there are two options. That somehow the CD4 T cells are contributing and interplay and interact with the cancer cell and or with the stroma. So first we wanted to see, is there a contribution of the CD4s with engaging with the CD8s? And so we generated tumor cells that we edited for deletion of MHC class 2. So these cancer cells cannot express MHC class 2 epitopes. And we exactly had the same effect. So MHC class 2 and therefore CD4 T cells do not have to target the cancer cell directly in this context. So then we said, okay, if it's not the cancer cell that is part of this game, then what about the stroma? And one experiment that we did was we depleted CD11C positive cells within the tumor by using DTR mice. And what we found was when you deplete CD11C APCs, positive APCs in the tumor, you completely lose the effect. So we knew that the CD11C APCs are critical for CD4 T cells to reprogram the CD8 T cells. So, but then the question was how? If it's not the numbers, but you know that the CD11C positive cells in the stroma are important, what is really the mechanism of how CD4 T cells mediate and help CD8 T cells to become effective cytolytic killer T cells? And that is an experiment that Gabby did, which I think was really a key study that really led us into understanding spatial positioning of T cells driving anti-tumor immunity. So he used 2 million of these tumor cells that I presented that express CD8 and CD4 epitopes. But then he said, let me mix 1 million of tumor cells that only express CD8 epitope or 1 million cells that only express CD4 epitopes. Let's mix them. And we made the model in a way that so basically you give exactly the same number of cancer cells, the same amount of antigen that is being presented in the lymph node and in the tumor. And we gave exactly the same number of CD8 and CD4 T cells in these two experiments, A and B, or cohorts. And what we found was really striking. Only model A, where you have the expression of CD8 and CD4 epitope on the surface of the tumor cell, the tumor got rejected. But the mixing experiment, where you give exactly the same number of T cells and antigen, but presented on different type of, or expressed on different type of tumor cells, there the model breaks apart. And when we looked within the tumor, again, before the tumor decides to regress or progress, we had exactly the same numbers of CD8 and CD4 T cells. So clearly the numbers do not predict whether the tumor is being rejected or continues to grow. So that led us to the hypothesis, it must be something about spatial positioning and spatial orientation within the tissue tumor that really determines anti-tumor immunity. And indeed, when we looked at this tumor that grew out through this mixing experiment, we saw patches of either CD8 expressing tumors or CD4 expressing tumors. So they were really mosaic. So that led us to hypothesize there is potentially a real important role of how CD8 and CD4s interact within the tumor. We came up with the hypothesis that is presented here. In the model A, where we ultimately have tumor regression, you have a cancer that expresses CD8 and CD4 epitopes. Therefore, every APC in the tumor will co-express and present on the surface CD8 and CD4 epitopes. That physically brings CD8 and CD4s in close proximity. CD4s can actively reprogram the CD8s, and therefore the CD8s become reprogrammed, go to the tumor and kill. In the other model, you have the where you have either a tumor cell expressing CD8 or CD4, you never have the expression of both epitopes on the same APC. Therefore, the CD4s cannot go together with the CD8s, and therefore there is no reprogramming possible, and that's what ultimately drives tumors to grow out. So how do you prove this? That really the 3-cell cluster is required for effector function in the tumor during the effector phase. We looked in the literature, and there was one paper published in 1987 by O'Malley and Mitchison, where they first proposed this idea of a 3-cell cluster being important for cytotoxicity in vivo. And this was an allogeneic transplant model, where these authors demonstrated that you really need a 3-cell cluster, a triad, that allows ultimately CD8 T cells to become cytotoxic and eliminate target cells. So we looked in the tumor literature, and we really didn't find whether there are triads required within tumors in order to be mediating tumor regression. And so Gabi remodeled the system by color coding each component and giving it a unique color. So the dendritic cells, the CD8s, the CD4s, and the tumor, and he did confocal analysis. And what he found indeed, that ultimately the tumors that regress had much higher numbers of triads compared to the tumors that ultimately grow out, but had equal numbers of CD11C YFP positive cells. So that really led us to hypothesize what is driving anti-tumor immunity and allows CD8 T cells to ultimately kill is really the formation of the triads. So we also did this not only for adoptive T cell therapy, we did this also for immune checkpoint blockade in our model, and we have exactly the same outcome, that when you are allowing the tumor to have triads, you actually lead to anti-tumor regression or tumor regression. And if there is an impairment in triad formation, then the tumors continue to grow. So now the question is, how do you really prove this has to be one and the same cell? We did a mixed bone marrow chimera, where we use bone marrow which where either MHC class 1 or MHC class 2 is knocked out. You mix them together. So you will never have a cell that expresses either MHC1 or MHC2 together on the surface. And obviously then control wild type bone marrow. And this is where really the striking finding we had, where the mixed bone marrow chimera is not controlling the tumor and only the wild type, suggesting it is really one cell that is mediating, or that the CD8 and the CD4s have to be on one cell in order to drive or this reprogramming and CD8 mediated toxicity. So now the last two slides, I'm going to just present a glimpse into the clinical data. So this was an amazing collaboration triggered through Matt, who introduced me to Hyung and Brian from the Baylor College of Medicine, who generously allowed us to use unpublished data from their cohort to ask the question, is triads associated with anti-tumor immunity also in the context of human solid tumors clinical responses? And Hyung and Brian, they had mesothelioma cohort patients, 15 patients where they were treated with immune checkpoint blockade, and then they did pre and post, during and after resection biopsies, and did this amazing multiplex image IMC imaging mass cytometry. And we went to them and asked, well, what is the predictive, the marker that could potentially demarcate your responders from the non-responders? They looked at CD8 infiltration, they looked at CD4 infiltration, and they looked at diet. So the specific CD4, CD11C or CD8, CD11C combination, and nothing really was holding up. The only feature that could demarcate responder from non-responders was really the formation of the triads. And when they specifically then looked in the triads, in the 3-cell clusters, what is the phenotype of these T cells? They are highly PD-1, RO, so they're clearly tumor reactive or activated. They make much higher interferon gamma, granzyme B. Here it's quantified. We also looked at the CD4 phenotype, and what was really nice to see, the CD4 T cells that are part of the triads have no or very little Treg FOXP3 expression, and only the TCD4s in the microenvironment and part of the diets are really having this FOXP3 CD4 feature. And there, this is the data set from the Baylor group, but a beautiful paper last summer from Alice Camphorst and Miriam Marat published niches and clusters within hepatocellular carcinoma patients, responders and non-responders, where they see similar features of CD4 T cells together with antigen presenting cells. And then a recent paper just last month from Nihar Cohen's group, also a beautiful these immune hubs where he demonstrates CD4 and CD8s and APCs together. So I think looking more at these human tumors, what are the spatial characteristics of those tumors and patients that respond or don't respond will be very exciting in the future to study. And with that, I'm going to close. We think that the CD4 T cells have an additional role to actually be required in tissues for mediating cytotoxic effector function of CD8 T cells. This is the model that we are hypothesizing. Now the question is, what are the signals? Is this a receptor ligand engagement that allows the reprogramming? Is it cytokines that are being released and really part of this micro environment and this niche? How can you engineer CD8, CD4 T cells that are cooperating for adoptive T cell therapy? How can you enforce triad formation in immune checkpoint or vaccinations and so forth? Those are the ongoing questions that we are currently addressing in the lab. And with that, I'm going to close. This is the last slide. The tumor, coming from the tumor context, really understanding what is derailing T cell differentiation in the context of tumors. How can we reprogram them? Really learning from the autoimmune context where things work too beautifully, where you have CD8 T cell continuously driving tissue destruction. Appreciating that the tissue draining lymph node might be the headquarter where the fates are being determined, but also realizing that within the tissue itself, during the effector phase, there might be unique spatial compositions and interactions, cell-cell interaction that really drive CD8 T cell cytotoxicity. And with that, I'm going to close. I would not sit here and talk to you without the amazing support of my terrific team This is an earlier version. This is a later version. Being a proud PI, amazing people in the team, numerous collaborators at MSK, outside of MSK. Without them, this work would not have been possible. Funding, and thank you for listening and having me.
Well, thank you very much, Andrea. I'm sure there's a lot of virtual applause, which you can't quite hear. I'm sure there will be plenty of questions, but of course, these will not be live. They will be via Twitter for all attendees. Here are the instructions. Just find the tweet that states ask questions for Dr. Andrea Schietinger here, and then you reply to that tweet with your question, and don't forget to mention hashtag Global Immuno. And so it just remains for me to thank Andrea once again for a superb talk. Thank all of you for attending and for your support of the Global Immunotalks seminar series. And I hope to see you all at our next seminar. And of course, if you want to revisit the present one, it will be posted on YouTube very shortly. So thank you all again. Thank you, Andrea, and enjoy the rest of your day wherever you happen to be.
Thank you. Bye-bye.